the epigenome changes across the life course. I think we've now profiled just over 1,500 brains. It's a slightly out-of-date slide. So this is showing data for about 1,400 brain samples. And this is for DNA methylation. Again, this is just highlighting one of these co-methylation modules and highlighting how that modular co-methylation is changing across the life course. And you can see the very dramatic effects that are occurring during the prenatal period. But what you also see is then more subtle effects occurring across the rest of the life course, but also very different effects in different regions of the brain. One thing I just want to point out here is that there's this kind of period in the late stages of pregnancy and also early life, childhood through to early adolescence, that still really remains a bit of a black box. We don't, because of the limited number of samples that are available from this period, this potentially very critical period remains a kind of window that we know very little about in terms of gene regulation, and I think that's something that we need to address moving into the future. But of course, what this allows us to do now that we have developed these patterns of normal, or inverted commas, normal development across aging and the brain, it allows us to start looking at some of the phenotypes that are not only associated with neurodevelopment but are associated with diseases that occur in old age. And I just want to touch a little bit on some of our work into Alzheimer's disease, because I think, again, these are some of the kind of most robust findings I think that we've generated from our work in epigenetic epidemiology. So I think you all know what Alzheimer's disease is. Obviously, progressive accumulation of neuropathology in the cortex, really characterized at a pathological level by these extracellular deposits of amyloid and elevated hyperphosphorylated tau, which obviously leads to the neurofibrillary tangles. One of the interesting things about Alzheimer's disease, it's a very progressive disease. It starts off really in the entorhinal cortex, hippocampal region, and spreads progressively through the cortex. But there are regions of the brain that remain very protected against neuropathology, like the cerebellum. And of course, Alzheimer's disease, in some cases, has a very early onset and familial causes, and this is caused by rare, very dominant mutations in genes like APP, presenilin 1, presenilin 2, and the MAPT gene. But most cases of Alzheimer's disease are much later onset. They're what we classify as sporadic Alzheimer's disease. It's still a strong genetic component to these. We know this from heritability estimates. And of course, GWAS studies have highlighted genes like ApoE and a range of other genes associated with Alzheimer's disease. Just as in schizophrenia, there appears to be this kind of polygenic burden associated with risk of schizophrenia. But the environment also plays a very important role. So we've undertaken many studies looking at Alzheimer's disease brain, and particularly in the context of DNA methylation, find very robust epigenetic signatures associated with disease in the cortex. So these are two papers that we were published back-to-back in Nature Neuroscience, I think about 4 years ago now. And the kind of really exciting thing here was that these two independent studies really homed in on exactly the same changes. So the samples themselves were very independent, but we find very strong evidence, particularly around this gene called ANK1, for evidence for differential methylation associated with disease. And this is a really robust finding. We've done several other studies since that have highlighted epigenetic variation within this gene associated with Alzheimer's disease. So I think this is a nice example of how these studies can work, and there's now a lot of kind of functional biology work that's come out of that's now really kind of pinpointing on that as a new mechanism in disease. This wasn't a gene that had been previously highlighted from GWAS studies. Just to kind of show you how reproducible these effects can be when this sort of work actually is finding real findings on the right is a scatter chart of the effect sizes between the two biggest studies to date of an EWOS of DNA methylation in Alzheimer's disease. So on the x-axis are the findings from our study. Each dot is a site that we find to be differentially methylated in Alzheimer's disease brain, and on the right, on the y-axis, is the data for the same sites in a much larger study from Harvard in the United States. And you can see, I've never seen such a concordance of effect sizes across studies. So this makes us really believe that these results are robust and reliable. The two red dots are the sites in ANC1 that we find associated with disease. The point I want you to take from this is that these changes are all cortex specific. So from each individual, we not only profiled the entorhinal cortex, this is the region of the brain that we know is implicated early on in disease, we also profiled other bits of the cortex, and we found the same signatures there, but they were somewhat attenuated. But if you took a region of the brain that showed no pathology, like the cerebellum, we didn't find any of these differences. And we also had blood samples from the same individuals taken before the individuals died, and again we didn't see any of the same changes there. So these changes were specific to the brain region involved in disease. Following on from that, we've highlighted other regions of the genome very robustly associated with Alzheimer's disease. This is a region spanning the HOX-A gene cluster. This has just been published if anyone's interested. But we've also started to look beyond DNA modification. So as I said earlier, DNA methylation is only one of a whole kind of suite of different modifications that act to regulate gene expression. And a lot of our focus is now switching to other types of modification, particularly certain histone modifications associated with active gene expression. And we're particularly interested in H3K27 acetylation. This is a robust marker of active enhancers and promoters. It's known to be strongly correlated with gene expression and transcription factor binding. So I just want to give you kind of one story about this modification. Why are we interested in it? As I said, it's important in marking active regions of gene transcription. It's relatively independent of other histone marks. Importantly, it shows inter-individual variation, so there's no point in studying a modification if it doesn't show differences between individuals, because obviously that's needed to associate that with disease. And in the context of Alzheimer's disease and actually other CNS diseases, histone acetylation is a really nice target to look at, because we know that histone acetylation modifiers are really under great interest for pharmaceutical companies as novel targets for disease. So there's previous evidence have suggested things like HDAC inhibitors have some kind of therapeutic benefits, not only in Alzheimer's disease, but certain types of cancer and other CNS disorders. So I'm just going to talk you through the results. This is just published in Nature Neuroscience a couple of months ago by Sarah Marzi, who's a PhD student in our group. And she took these same samples that we'd profiled DNA methylation for and undertook highly parallel chip sequencing for H3K27 acetylation. This heat map just really highlights the results. And again, I think it just shows you the power of looking directly at these molecular readouts in disease-relevant regions of the brain, because you can almost perfectly characterize cases from controls based on their H3K27 acetylation profile. On the left are regions that are hypoacetylated in Alzheimer's disease, on the right regions that are hyperacetylated. And the other kind of really interesting thing for me here was that, so these are all, the samples that we profiled were all late onset sporadic Alzheimer's disease cases. None of them harbored the familial mutations in genes like tau, APP, presenilin-1 or presenilin-2. But the changes that we saw at a mechanistic level in terms of gene regulation were all within these genes, these key genes that are involved in early onset Alzheimer's disease. So again, it's showing that there are, as people have hypothesized, very convergent mechanistic similarities between late onset and early onset Alzheimer's disease, but it appears that maybe the dysregulation of these genes in late onset Alzheimer's disease may be via some of these epigenetic modifications rather than pathogenic mutations within the protein region of the gene itself. And the other message from this is that, again, these changes were all cortex-specific, and they particularly occurred in cortex-specific enhancer domains, again telling us that looking at the relevant piece of the brain is likely to be very important in identifying these changes. And this brings me on to the next section of the talk, and this is really just to touch on some of the issues of doing this type of work. And you'll hear more about this in the workshop in the coming days. But if you think about it, doing genetic epidemiology is relatively easy to do because all you need is a blood sample. The same genetic sequence is present in every cell that you look at. It doesn't matter when you take that blood sample for genotyping because you're born essentially with the same DNA sequence as you die with. It doesn't matter what you do during your life because that isn't going to change your DNA sequence. The disease itself doesn't change your DNA sequence. And of course, we know a lot about the DNA sequence. It's been sequenced, there's a whole catalogue of polymorphic variants, and there's lots and lots of data that we can refer to in order to identify genetic variants associated with disease. And it's worth bearing in mind that none of that is true when we think about doing epigenetic epidemiology, and in fact, probably molecular epidemiology in general. And at the moment, many of the studies, including many of our own, so we're as guilty of this as anyone, are really still very based on very small numbers. They often use the wrong tissues or cell types, and the study design that's used is often incorrect. I'll talk a little bit about that in a minute. And of course, there's a lot of interest in this, as evidenced by this workshop and many others. A lot of the popular media is really jumping on the importance of epigenetics, but we have to be really careful in managing the kind of hype and over-interpretation that's often kind of put on epigenetics as playing a role in diseases. And if anyone's interested in reading more about that, this is a kind of opinion piece that I wrote with Bas Heijmans in Leiden, who's another epigenetic epidemiologist a couple of years ago. So I just want to give you one example of why it's very easy to be misled. So this is a big study of schizophrenia that we did using peripheral blood tissue, about 1,000 cases and 1,000 controls, and this is the Manhattan plot that we got after doing this comparison. And you can, just as in a GWAS study, basically you get very excited by anything above the red line because that's things with a genome-wide significance to survive correction for multiple testing. And we were interested because these sites kind of mapped onto genes that had functions that might well be important or interesting. And we looked, so we started to kind of look into this a bit more, and we realized that this Manhattan plot that we found for our schizophrenia study was a total mirror image of this Manhattan plot here. And what this is a Manhattan plot of smoking. So this is a Manhattan plot of tobacco smoking. And this shows how easy it is to be misled in this type of work, because we know that schizophrenia patients smoke at a much higher rate than non-schizophrenia patients. And so what we'd inadvertently done wasn't really find differential methylation associated with schizophrenia. What we'd identified was differential methylation associated with tobacco smoking. And so it's important to realise that the study designs that have been used for genetic epidemiology are really unlikely to be optimal for molecular epidemiology. And a big mistake is to move simply from profiling the same samples that have been used for GWAS studies, for instance, and putting them through all the latest EWAS arrays, because you'll end up with huge confounding like that. So all isn't lost. Because actually the effects of some of these environmental exposures are so great that we can actually start to use that data to our advantage, and we can actually start to infer exposures from the DNA methylation data itself. So this is what we call a smoking score, basically derived from an EWAS of smoking. This is work by Eilish Hannon in the group, who incidentally was supposed to be here with us today, but fortunately for her had a baby, but unfortunately for her couldn't come to India. But what this shows is how this smoking score, so DNA methylation at the smoking score, can almost perfectly differentiate between smokers in the red and non-smokers in the green. And in fact, if I was to take a DNA sample from people in this audience, I think I'd do a pretty good job just from the DNA methylation profile of working out who was a current smoker and who wasn't. And just to show this, I think has real important public health messages as well. So this is just a figure from a paper that we've recently published. This is working on neonatal blood spots from the Danish National Health Register. It's a really great resource. We've profiled about 1,500 of these samples at birth, and then you can link this to all the records, public health records known about these individuals. And one of the things we looked at was whether the mum had reported smoking or not before birth. And there was, again, this is just a box plot of the smoking score in the offspring. Bear in mind, this is samples taken at 2 days of age. It's a little heel prick blood test. This is a differential methylation between individuals whose mum smoked compared to those that didn't. So you can see that the maternal exposures have very dramatic effects on the gene regulation in the offspring. I think that's an important message. So one of the other issues that we have to deal with when we're working with a tissue like blood is that there's a lot of cellular heterogeneity. So this is obviously the brain. We know there are many different cell types in the brain, but this is blood, and again, many different cell types in the blood. And of course, as I said earlier, each one of these different cell types, because it's a different cell type, will have a very different pattern of gene expression, a very different epigenetic profile. And one of the important things to be wary of in epigenetic studies or gene expression studies using blood is that many of the many factors that may be associated with disease, such as infection, age, stress, diet, exercise, a whole list of things, can actually act to kind of shift the blood cell proportions in the blood. And so that the changes that are identified in studies of blood may often just simply reflect differences in cell counts rather than some underlying pathogenic mechanism. So again, there are ways around that, and Emma and others in our group are trying to develop ways that we can utilize data generated on bulk cortex tissue to actually work out the profiles seen in the individual types of cells within the brain. So we're using a kind of range of different antibodies that mark specific neuronal and other types of neural cell types in the brain in combination with FACS sorting. We're able to profile what we think are relatively pure populations of neurons, oligodendrocytes and astrocytes from a bulk portion of brain, and we're able to then use those purified cell populations to undertake our epigenomic profiling. And again, just to highlight why that's important, you'll see this is data from one individual. So you'll see the column marked bulk, so that's a profile from the Illumina 450K array, and then you'll see the profiles from the more purified cell populations, and you'll see that they're dramatically different. So that if you're looking at a disease like Alzheimer's disease that maybe is characterized by a loss of neurons, you could see an underlying change in the bulk tissue that has nothing to do with an underlying epigenetic change, but may simply be reflecting some of these changes in cell proportions. But again, we can use this to our advantage because we have these known profiles from pure populations. We can derive via some clever bioinformatic and statistical approaches ways of actually deconvoluting the cell type proportions from bulk tissue. And just to show that works, this is DNA methylation data again from our Alzheimer's disease versus control brains. And then what we've plotted here is the estimated neuronal proportion in our cases versus controls. And you'll see a very significant shift to reduced neuron proportions, as you would expect in Alzheimer's disease compared to controls. And we often, so we obviously always include these types of covariates in our analyses, and also include gene expression data where we have matched RNA sequencing data on cell type specific genes that enable us to be more robustly confident in the results that we see aren't simply reflecting cell type changes. Even when you have pure populations of cells, it's always worth noting that there's going to be a lot of heterogeneity even within those pure populations, and that's why there's a real move now towards single cell transcriptomics and epigenomics. And I think the technology here is still very much in its infancy, but I think this is something that's going to be really important in moving the field forward. So in just this hypothetical example here, you can see how a gene expression profile on a bulk tissue could really hide 3 different populations of cells that are seen within that bulk tissue, where maybe some are showing elevated expression and others are showing reduced expression. So one of the kind of limitations of this work, as I've highlighted, is that if it turns out that we need the numbers of individuals that have been used in genetic association studies, we might be really in trouble, because although in the UK and elsewhere we have access to very nice collections of post-mortem brain tissue, these obviously are in the nature of hundreds of individuals rather than maybe tens of thousands of individuals that have been needed to identify changes in GWAS. So one of the interests of our group is to highlight or examine the extent to which variation detected peripherally can tell us about variation in individual different regions of the brain. And if anyone's interested, this is a study where we took 100 individuals. We took brain tissue post-mortem and blood tissue taken immediately before death, and actually profiled the patterns of DNA methylation across these different tissues and brain regions, and examine the extent to which variation in blood mirrors variation in the brain. And the take-home message is that this works for some regions of the genome and doesn't for others. So I think it's a very important thing to bear in mind when making links between variation detected peripherally and variation that may be more pathogenically important in the brain. Of course, using blood can still be really important, because using blood is an easily accessible tissue. If we ever want to derive biomarkers, it's going to have to be in such a tissue. And of course, understanding the consequences of disease may well be just as important as understanding the actual causes of disease. It's just worth, I think, framing your question within the context of the tissue that's available. And so we are continuing to do lots of work using blood because we have access to big data sets using blood. This is our latest schizophrenia case control sample now. We're up to about 4,500 individuals. This is data. Many of these are data sets that have been included in the big PGC GWAS study that we're now profiling DNA methylation data on. I just wanted to kind of give you a few kind of headline findings from this. This is again all unpublished work, but again you can see that our smoking score seems to really differentiate between cases and controls. So in every single cohort that we've looked at, there is elevated smoking score in the schizophrenia cases compared to controls. So it's really important to control for that covariate in the analysis. We also can look at different cell types, because just as in brain we can deconvolute different cell types, you can do the same with the cell types in circulating blood. And again, here we see very different cell type proportions in cases compared to controls. One of the really interesting ones, I think, relates to granulocytes. That seems to be different in all our case control populations, other than our 2 cohorts that are focused on first episode psychosis. And I think the nice thing here is that obviously we know that many of the medications that are used to treat schizophrenia alter some of these blood cell proportions, and so it's interesting that you don't see that in the non-medicated first episode psychosis samples. So again, we've done a whole genome methylation profiling in this big sample, meta-analysed the results across the different cohorts, and find almost 400 regions of the genome harbouring differential methylation in cases compared to controls. I won't labour on the results too much. This is just the top differentially methylated position. You can see the forest plots on the bottom showing very consistent but relatively small effects across all the different cohorts. Interestingly, this is a a CPG site in the promoter of a gene called ORTS2, which, as by its name, you can probably understand that this has been previously implicated in autism. And this is the list of all the top ranked differentially methylated sites associated with schizophrenia. I've highlighted the ones in red because, again, I want to highlight the overlap that we see between our epigenetic results and genetic results. The ones in red are all genes that have been implicated from genetic studies of schizophrenia. And again, just as in our Alzheimer's results, these are really consistent and reproducible effects. So this is just a couple of scatter plots showing the high reproducibility of our schizophrenia-associated differential methylation in our discovery cohorts on the x-axis and a couple of replication cohorts on the y-axis. And you can see a very consistent correlation of effect sizes that make us believe that these are at least robust signatures of peripheral blood signatures of schizophrenia. I've highlighted MZ twins here. Emma's going to talk about the MZ twin results in her talk next. I think those are particularly interesting because twins are a really powerful tool for doing molecular epidemiology because they allow us to control for many of the confounds that I've talked about. So they obviously share the same genotype, they've got the same parents, we can collect blood at exactly the same age, they're the same sex, and to a certain extent they've shared the very similar prenatal and immediate perinatal